Hello, everyone, and thank you for joining us today. My name is Kira Mills from Samba Scientific, and I'll be the moderator for this webinar. Optical genome mapping, or OGM, enables the detection of large structural variations associated with complex diseases such as cancer and genetic disorders that are often missed with traditional short-read sequencing techniques. OGM leverages high-quality, ultra-high molecular weight, or UHMW DNA, as input, but most nucleic acid preparation methods produce highly fragmented molecules. Bionano's Ionic system automates the gentle purification of UHMW DNA ideal for use in OGM. This session will feature early use case data from two researchers, Dr. Nikhil Sahajpal, an LGG Fellow at Greenwood Genetic Center, and Dr. Kornelia Neveling from the Department of Human Genetics at Radboud University Medical Center in the Netherlands. Additionally, we are joined by Bionano's Vice President of Clinical Affairs, Dr. Alex Hastie. In this webinar, you'll learn how researchers have successfully integrated the Ionic system for automated UHMW DNA extraction into their OGM workflows, achieving faster, more efficient results, as well as a deep understanding of the Ionic system and the Stratysphere workflow, and discover how this application-agnostic solution can streamline sample preparation across various genomic applications. Now, without further delay, I will turn it over to Alex.
Hello, everyone. Welcome to today's webinar. Bionano is excited about the tremendous progress that has been made with the development of an automated DNA extraction system that will become a key part of the Stratysphere for OGM workflows. My name is Alex Hastie, and I will provide an introduction to the Bionano technology and introduce two amazing speakers who will discuss their experience with the Ionic system. Today, we realize that we're still not finding all of the variants that matter in genetic disease. Across indications, the figure varies, but on average, we find meaningful answers in only about half of the samples. So how do we find more genomic variation that directly connects to answers in human disease and eventually better care? In other words, how do we start to unpack the other 50%? After several years of a growing body of literature demonstrating the tremendous value of optical genome mapping in many disease workflows, international consortiums like the Gregor Consortium and others are proposing new workflows that using state-of-the-art technologies. And that includes optical genome mapping in many of these workflows. Over the last 3 years, we've seen an explosion of publications demonstrating the important progress that clinical researchers are driving. We've seen important progress that clinical researchers are driving that demonstrates the tremendous potential value of optical genome mapping in clinical practice. This is acutely exemplified by the sharp increase of unique clinical research subjects published in the literature, which has now reached almost 7,000 cases. Genomic variants come in every size, from one base pair to whole chromosomes, and this continuum plots this size range is plotted on this slide. We know that short and long read sequencing can identify small variants with high sensitivity. And traditionally, cytogenomic technologies have been used to identify large variants. These include FISH, microarray, and karyotyping. All of these cytogenomic methods have limitations of their own, and so they must be used together in many cases. The reason that OGM is now included in essentially all genetic disease research is that it not only simplifies the cytogenomic workflow, karyotyping, FISH, and chromosomal microarray, and increases the sensitivity for those variants, it also bridges a major gap between cyto and molecular techniques. So this slide shows essentially how optical genome mapping works. DNA molecules are shown in this video flowing through a nanochannel array. The instrumentation, the Sapphire or the Stratus instrument, will electrophoresis DNA into the nanochannel array and thereby linearizing that DNA. Once the DNA is linearized, the electrophoresis can stop and the DNA can be imaged. The images of these DNA molecules serve as a barcode for the genome and allow the identification of variants in the genome by comparing those barcodes to the reference genome. The OGM workflow consists of a 3 to 4 day turnaround time from sample to answer. And this is completely supported by Bionano. Briefly, DNA is isolated and labeled using Bionano prep kits. Data is collected on a Sapphire instrument, or today, a Stratus instrument can be used as an alternative, as a high throughput alternative. The instrument will electrophorese the DNA as you saw in the previous slide and image those molecules. The molecules are extracted and compared to a reference genome through algorithms that detect variants and display several different useful visualizations of those variants. Bionano has also implemented a tertiary software that enables the efficient analysis of variants for their relationship to the biology of the sample undergoing research. Today, we have manual DNA extraction kits called ultra high molecular weight DNA SP kit that is used to extract DNA for labeling with a direct label and stain kit, and then loading onto the Sapphire or the Stratus instrument for data collection. Today, we're going to hear much more about the Ionic system, a new automated system for gentle extraction of automated gentle extraction of high molecular weight DNA that will feed into the optical genome mapping workflow. The key advantages of the Ionic purification system are that it is an automated sample purification with limited hands-on time. It has high yield and gentle process to maintain intact DNA. You can run 8 samples at a time on this automated system, and it has a fast turnaround time from sample workflows of 2.5 hours. This involves the instrumentation, a benchtop system, and consumable chips. So this video shows the process of the purification of the DNA on the Ionic system. We're actually seeing the DNA in green moving through a buffer where it's being separated from impurities. And finally, the DNA is deposited into a well where it can be pipetted out and added to your next steps of the procedure. We can see it's just collected in a well at the end. I also want to provide a little bit of information about additional steps in the Stratysphere workflow. With the launch of the high throughput Stratus system, we've increased the capacity of optical genome mapping in labs. This system has a high capacity with containing the ability to load 12 samples at a time. And these samples are all independent. So a sample, when it's finished, can be replaced with a new sample at any given time through random access of the chips. That means that there's no batching required, and samples can be sample batching can be optimized for the lab's needs. It also includes a jump to queue feature to prioritize certain high priority on-demand samples. This will jump the new sample, the priority sample, to the front of the queue. And all of this comes with reduced turnaround time and higher throughput, about 4 times the throughput of a Sapphire system. And I just wanted to briefly highlight our new analytic software, tertiary software, VIA. This is an integrated software which enables the analysis of optical genome mapping data alongside NGS and microarray data. And this software integrates many databases, as well as the ability to set up customized decision trees to automate the reporting of results from individual samples. Based on external user experience with the two customers who we'll hear from today, we are encouraged by what we've seen for Ionic as a streamlined workflow for ultra-high molecular weight DNA isolation. The next steps for commercialization will involve gaining more external user feedback through additional evaluations and finishing development and commercialization. While we don't expect Ionic to be available in 2024, we do expect the commercial release in the future, and we'll update everyone as the timing of commercial release becomes more definite. Now I'd like to briefly introduce our amazing speakers and my friends, Nikhil Sahajpal and Kornelia Neveling. Nikhil will speak first. He is currently an LGG fellow at the Greenwood Genetic Center, where he is training in laboratory genetics and genomics and conducting research on optical genome mapping, among other things. Previously, he led OGM research in Ravindra Kole's lab at Augusta University. Following Nikhil's talk, Dr. Kornelia Neveling will share some of her work at the Radboud University Medical Center, where she has led some of the first studies demonstrating that optical genome mapping can serve as an alternative to classic cytogenetic methods. Today, she will focus on her effort to automate the production level OGM workflow using the Ionic system. And now, Nikhil, the floor is yours.
Thank you, Dr. Hastie, for the introduction. I'm excited to share our experience of using optical genome mapping and the Ionic DNA isolation purification system here at the Greenwood Genetic Center. But before we dive into that, I would like to introduce you to the Greenwood Genetic Center as well, if you are not already aware, with the intent of highlighting the involvement of the center with optical genome mapping research. We are located in a small town of Greenwood in the state of South Carolina, but the center has kept pace with the discoveries around the world. In context of optical genome mapping, the center would be amongst the first, if not the first, that explored optical genome mapping to investigate rare disease cohorts. And I would like to highlight just one of these cohorts, which is the neural tube defect cohort. Data on over 100 cases were collected back in time on the older instrument, which is not the Sapphire as we are talking about Stratus, but the older instrument, which was Iris. And that data that was collected back in time was reanalyzed because of the improvements in the software, the variant calling, and the variant filtering abilities. And we were really excited about the results. So we identified causal findings in 8% of these cases and additional candidate findings in 22% of these cases. Anyone who is following neural tube defect research would, I believe, share our enthusiasm and excitement with these findings. The manuscript with this cohort is under revision in the peer review process, and I hope that it will be available for everyone to read. But all to say that this really motivated us, and there's a cohort of additional 200 cases that is in progress with data collection and analysis on using the optical genome mapping software. I would also like to highlight that the center is part of several multi-institutional studies. These include studies that are evaluating rare disease cohorts or constitutional postnatal cases, or the studies or groups that are evaluating optical genome mapping for prenatal genetic testing, or be it collaborations that are evaluating the use of optical genome mapping in hematological malignancies. So you can imagine with this experience, we very recently launched clinical optical genome mapping research essay, which is available in two different forms or in two different workflows. One is the optical genome mapping complete essay, where we look at unbiased genome-wide SV analysis throughout the genome, whereas the other one is optical genome mapping targeted, where we investigate a certain region of the genome or a specific gene of interest. So with this, I'll move to the topic of interest for today's discussion. This cartoon here depicts the entire optical genome mapping workflow, as was just briefed by Dr. Hastie, where we have the isolation of high molecular weight DNA on day 1. And on day 2, the isolated DNA is labeled and then processed downstream for data collection on the instrument. Now, if you ask me what's the most important step in this protocol, I would say it's the DNA isolation. And the reason I say that is because the assay is primarily based on high molecular weight DNA. It's a very robust process, although it is manual. And I'm saying this with some level of personal experience because I've isolated DNA on more than 700 cases, but there are certain challenges with the current isolation method. So if we look at the current challenges that we face with the current DNA isolation method, obviously because it's a manual process, there is tech variability. There's a requirement of certain care that needs to be exercised when one is isolating DNA, because if you're harsh during the DNA isolation steps, it can fragment the DNA and would not result in the ideal DNA size that is required for this assay. Another point here is that the current DNA isolation method is based on a nanobind disk method, where the precipitation and cleaning of the DNA happens on a nanobind disk. And although this does not happen very often, there is a chance of losing the DNA. So at the end of the DNA isolation protocol, one might end up with no DNA because you can lose the DNA if it is not properly precipitated on the bead, the disk, or if it's not, it's lost during the washing steps. Another important point here is that there's manual washing of the DNA. So there can be a chance that the contaminants pass on during the isolation procedure. So this can cause issues during the labeling and data collection. Also, once the DNA is collected, there are challenges with respect to the homogeneity or the viscosity of the DNA. Many of you who are attending this webinar and are performing this DNA isolation would be aware that at times the DNA is very viscous. It's not homogeneous in the eluate. So this can pose challenges during the labeling procedure and during the data collection. And also at times we need to give more time for the DNA to homogenize. So this can impact the turnaround time or the deadlines you are trying to meet. So there are certain challenges with respect to the current DNA isolation procedure. And I know that we are discussing the Ionic DNA isolation purification system, but this is not the reason for highlighting these challenges. The gravity of these challenges is real to an extent that we actually wrote a troubleshooting guide. And for those of you who are interested, I encourage reading this paper if you are facing any challenges with the current DNA isolation workflow. And if you'll trace back to any of the analytical or post-analytical QC metrics, if those are not met, then you would trace it in many instances back to the DNA isolation protocol. So I'm not going to go over this entire table, but would say that if the DNA is clean and is high molecular weight, then the chances that you do not meet those QC parameters, the chances of that is very low. Now, the Ionic system gives us an opportunity to alleviate these challenges. So we evaluated the Ionic system using 48 different samples. These included control samples and also included well-characterized samples with known SVs. The evaluation parameters included DNA yield, eluate volume, and homogeneity. And I'll emphasize that across the 48 samples, we saw very uniform outputs or measurements with respect to the DNA yield, the eluate volume, and especially I would like to highlight the homogeneity here, that the DNA was really homogeneous. It was ready to be prepped for the next step. Then we label these DNA, the isolated DNAs from these different samples, and process all the way through data collection, and looked at the analytical QC parameters, which we all are well aware of: the average size N50, the labeling density, the map rate, and the throughput. And finally, we also compared for variant concordance between the DNA that was isolated with the Ionic system and the DNA that was isolated with the conventional SP method. So what I'm sharing here is the analytical QC parameters. The bars that you see in different shades of blue are samples that were isolated with the conventional SP system, and I've done random sampling of our database to pull these numbers out. And the bar in green that you see is the one with the Ionic isolation method. So the average N50, the minimum threshold is 220 kilobase pairs. You can see the N50 with the SP method is way above the threshold. The N50 with the Ionic method is 240 kilobase pairs. But I would like to say that the range is very narrow within that 240 kilobase pair range, and we'll see how this can be advantageous with respect to the downstream steps and data collection. The minimum map rate threshold is 70%, and you can see that it is way higher with all the different isolation methods. The average label density needs to be anywhere between 13 to 17 labels per 100 kilobase pairs, and you can see that the samples with the Ionic method sits right at the mean of 15. And most exciting of these, and probably an expected result, is the throughput. So this is how fast the data is being collected on the system. And as you can see that it is the fastest for the Ionic method. And this makes sense because the data collection is actually a function of the purity of the DNA and the size of the DNA. So if the DNA is not pure, it can clog the nanochannels. And also if the size is too large, it can clog the channels and can impede the data collection. So that having that tight N50 size range is critical to also data collection. Now, we also measured the concordance of different SVs, and we chose some difficult to map regions. One of these would be the FSHD, and you can see the top graph is from the SP isolation method, and the bottom is from the Ionic method, and you can see the concordance between these two procedures with respect to the final variant calling, where we see the two 4QA haplotypes and a repeat count of 2 and 26 with both methods. And I'm not showing the SP variant calls here, but I would like to mention that we did include different variant classes such as SIGDUP-mediated duplications, interstitial deletions, and terminal chromosomal deletions, such as this shown here in this image. And all to say that all the variants were concordant between the Ionic method and the SP method. So coming back to the entire workflow, what we have here is that we are replacing the manual DNA isolation with a semi-automated Ionic method, and this provides us with a very uniform pure high molecular weight DNA. And what it possibly provides us as well is the possibility to consolidate the entire workflow in one day. And we can talk more about that, but it's a play of how homogeneous that DNA is that this really is a possibility now to consolidate from a 2-day workflow to a 1-day workflow. So just to conclude, the ITP or the Ionic method provides us an homogeneous DNA isolation purification method. It's much faster than the conventional method. It's about 2 hours if you take the entire time from pulling the samples to the final eluate being collected in the tubes. For blood samples, for cells, it's 1.5 hours. This is when if you already have cells pelleted sitting in the -80, you can add the cell counting time to this 1.5 hours if you have not already done that. In our hands and in our experience, there was 100% samples met the QC parameters. 100% variant calls were concordant for different SV classes. And as I mentioned, there's a potential to consolidate the optical genome mapping workflow in 1 day. And the isolated DNA is compatible with other long read, short read methods. At least the ones that we have tested are the nanopore sequencing and Illumina short reads. But I do not see any problem with that it would possess for other methodologies. So this is all I have, and I'll turn over to Dr. Neveling.
Here at Radboud UMC, we got our first Sapphire instrument in 2019. And the main question back then was whether OGM would be able to replace classical cytogenetic techniques. As said, we started 2 proof of principle studies, one with a cohort containing 85 samples with constitutional aberrations, and the other one with 52 samples with hematological malignancies. These are retrospective studies, so meaning all of these samples have been analyzed by standard of care previously. And when we compared the data, we saw that OGM was able to identify all variants that had previously been identified by standard of care, meaning we had a concordance of 100% for both of these studies. And that was, of course, excellent data. So we were super happy and published these studies back-to-back in the American Journal of Human Genetics in 2021. And based on these data, we went on and directly performed the next study. And this was a study containing 50 acute myeloid leukemia samples. And these were now done in a prospective way, meaning that also here we did a standard of care being FISH, karyotyping, and CNV microarray for all of these samples. But we compared the data afterwards. So we did not know what to expect when we analyzed the OGM data. But when we then compared, we saw that again, OGM was able to identify all variants that had been seen by standard of care as well. But OGM detected more. So we found in another 10% of the samples clinically relevant findings that were not detected by standard of care. And in another 10%, we got more details on the aberrations that were found by standard of care. So think about maybe a fusion partner that we're now able to identify. Then this was used as a clinical utility study. So we tested a lot more things than just looking at the data. And I'm just highlighting one of those here, and that was a turnaround time that turned out to be really significantly faster than standard of care. And this was mainly due to the interpretation time that was 1/4 faster compared to standard of care. And during that study, we realized already we cannot do that with one Sapphire. So we got a second system in September 2022. And based on all of these data, our management team then decided that we would like to implement OGM in clinical routine. And I'm super happy to tell you here that we succeeded. So now, since the 1st of October, we are offering a clinical routine using OGM for AML in our laboratory. And I hope that we can follow with different indications soon. But we are not using OGM only to analyze cytogenetic techniques or replace cytogenetic techniques, but we also use it to analyze rare diseases. And here we try to identify so far hidden structural variants. So think of exome or genome negative cases that we analyze by OGM. And in those, currently, we are identifying structural variants or pathogenic structural variants, I have to say, in a roughly 10 to 15% of cases. We have a lot of small research projects, like you see here, these two papers on the right side. But we're also involved in large projects, such as the European-funded SolveID consortium. We do a lot of singles and samples, but we especially like also patient-parent trios, where we look at the de novo approach. And this is very useful using OGM due to the very low false positive rate. Further, we are currently investigating repeat expansions, and we just have a paper on bioRxiv where we describe repeat expansion analysis for 85 samples with myotonic dystrophy type 1 and type 2 and Campbell syndrome. So looking at DMPK, CMPP, and RFC1. And these are only 3 loci, but actually we think that you could use OGM as a rather generic approaches for many more of these repeat expansion loci. And then there's a lot of other things you can do. So we saw, for example, that you could use OGM as a replacement for MLPA, especially for multi-gene copies where you need orientation and phasing or allelic information. And you can also use OGM as a standard QC2 just for iPSC cells, organoids, or CRISPR-Cas cell lines, for example. So I think you can imagine having all these routine workflow and research projects, having 2 Sapphires is just not enough. So here I'm happy to show you our 3rd machine. And I think you know these guys, Alex and Alka, standing in front of our new Stratus system. And I think you can appreciate, and Nikhil said that as well already, we really felt the need to automate. And therefore, a while ago, actually already, we started a 3-way collaboration with Bionano and Hamilton to automate the isolation of ultra-high molecular weight DNA on the long-string Vantage or STAR-E system. And this is actually up and running now. We can do both DNA isolation, actually also labeling already on that system, currently for bone marrows, and we are now working on different tissues for that. And this is what is currently used in our routine workflow. But nevertheless, we were, of course, super interested as well to have a look at the Ionic system. And this is what I would like to show you today. So the Ionic system, we heard that already, is a purification system that is used for extraction purification of both DNA and RNA from cells, tissues, and FFPE using a technology called isotachophoresis. So this technology is out for a while, but now there's also the possibility to use it for the isolation of ultra-high molecular weight DNA. And this is, of course, where we became interested. Alex said that already, so I'm not going too much into detail here, but isotachophoresis now separates and concentrates charged molecules in solution based on their electrophoretic mobility. And we do that on a chip, which is called the Ionic fluidic chip, where you can isolate 8 samples at a time. So you just add your lysed samples on that chip together with different respective buffers that you need. You apply an electric field, and then the molecules are moving through the chip and thereby separating. And once your DNA is coming to the extraction well, you just can pipette it out and go on with further analysis. So the advantage of that system is that the DNA is not bound to any surface, meaning there's no columns and no beads involved. And accordingly, you do not lose your samples or damage it when you try to get the DNA off that surface again. So there's no denaturation, no dehydration, no repetitive washing or whatever you can think of. So DNA that we get off is really better quality with especially lower fragmentation. If we compare the manual isolation with the automated one, manually, we heard that already in a talk from Nikhil, the high viscosity of this ultra-high molecular weight DNA, that is really tricky and makes samples not so easy to handle. And it requires really a lot of user experience. And now the Ionic is an automated system with less viscous DNA, so really less user experience is needed here. Concerning throughput, manually we currently do a maximum of 6 samples at a time, and for bone marrow actually we prefer to do 4, whereas I said already on a chip you can isolate 8 samples on one chip, and you can manage to do 2 chips a day. But I think what's maybe for us more important is that due to the super high viscosity of the DNA, manually we often need 1 to 3 days of homogenization before we can go on with labeling. Whereas on the Ionic, the DNA is less viscous and there's just 1 step of 1 hour homogenization needed, and then you can directly go on with labeling the same day. And finally, well, of course, a manual DNA isolation is much more error prone than an automated one, but it's not inherent for the Ionic, but this is just true for every automated process. This is just less error prone. So we decided that we would like to test the system, and we had a training in July this year. And here you see Evelyn and Rona, 2 super experienced OGM technicians, together with Valeria, who performed the training with us. We have isolated 56 samples, so that was corresponding to 7 plates of 8 samples each. And we have tested DNA isolation from blood, from cell pellets, and from bone marrow aspirates. And you see here in dark blue material that was provided by Bionano, and in light blue, this is material that was just from Radboud, our own material. We isolated the DNA on the chip as described previously, and then we saw 2 things that we found were remarkable. One of them I mentioned already is the DNA was really far less viscous compared to the manual isolated DNA. And the other one was about the qubit measurement. So usually we do 3 qubit measurements per sample, and then there's quite a variation in there. So we calculate the coefficient of variation, the CV, and depending on that value, we decide whether we can go on with labeling or not. And here for the Ionic, that was really surprising. The 3 measurements that we do for 1 sample are always nearly identical, and we had a CV of close to 0 for really all samples. But this is within 1 sample, so if you look across different samples, we do see here that also within a given tissue, the sample concentration was very close to each other, but we do see concentration differences between different tissues, and that may be, for example, due to different protocols. I don't know the reason, but still, it's very stable what we see here. We then took 48 of these samples. We labeled them manually and analyzed them on the Stratus. And the Stratus, you heard that already, comes now with a chip where you load a single sample on a chip instead of 3, what we used from the Sapphire. The chip runs much faster compared to Sapphires, and during the run, again, we can have a look at the QC value as a dashboard, and this is very similar to what we used from Sapphire. So for people among you who are used to look at QC values of OGM, you can appreciate, I think, that this sample we are looking at here is a really good QC sample. But then again, this is just a single sample, and we would need to see the quality of all of them. And I have summarized them in a following slide. And when we start with the N50, you see here in dark blue, the N50 larger than 150 kb. And in light blue, the N50 larger 20 kb. So these are the 2 values here we are mainly looking at. And I hope you can appreciate that for both of them, nearly all samples are far above spec, with a mean of around 300 for the N50 larger 150 kb, and a mean higher than 250 for the N50 larger 20 kb. So just for comparison, we wanted to know how that would look like on the Sapphire. And I have to say, this is not a one-on-one comparison because we had Ionic isolated DNAs on Stratus, but the samples on Sapphire are all manually isolated DNA, so please keep that in mind. But just for comparison, I took 50 random recent samples, and compared them. And again, you see here, these are very good quality QC samples, but still the N50 of Ionic Stratus combination is higher compared to the manual DNA on Sapphire. And we see that especially for the N50 larger 20 kb. So there's really a huge difference, and this is what we think is coming due to the ionic isolation because we have less degraded DNA. When it comes to map rate, we see excellent values for both of these data sets. The ionic isolated DNA on Stratus have a mean map rate higher 90%. But this is true for manually isolated DNAs on Sapphire as well. So we just see the map rate slightly higher for the Ionic Stratus combination, but both of them are perfectly fine. And finally, the label density, you saw that in Nikhil's talk already. Also here, perfectly within specs for both of these combinations, Ionic Stratus and manually isolated DNA on Sapphire. And the label density on Ionic Stratus is maybe a little bit lower compared to the other one, but both of them are perfectly fine. So overall, we can say here that what we've seen so far, that the performance of the combination of Ionic isolated DNA on Stratus is equal or even better compared to manually isolated DNA analyzed on Sapphire. And of course, we have also then analyzed the data, or we're currently analyzing these data, and this is now done using the guided assembly tool, which comes on Stratus instead of the previously de novo assembly tool and REP analysis that we used from the Sapphire system. So again, we have a different parameter here, and we have not a one-on-one comparison yet. But if you look at the overall number of structural variants, we can see that we get around 6,000 structural variants per sample. And if you then filter for rare variants only, we end up with around 60 structural variants per sample. And this is exactly the number that we also used from Sapphire. So also here we can see that, I'll say that overall the numbers of SD codes is comparable, although we have not done a proper one-on-one validation yet, which still needs to be done. So this is the data that we currently have, and how are we going to go on from here? Well, we definitely would love to go on further with the Ionic, so we really hope that we can get additional reagents. So we would like, for example, to test the lower input, and I just realized I forgot to tell you that we have started here with 1 million cells, so all the data are based on 1 million cells, but we would like to test if you can go even down, that would be great. And also we would love to test different tissues, for example. So think about saliva or buccal swab that have been very difficult until now. So it would be great to see how they would perform with the Ionic system. And then there's additional things, of course, that we can think of that we would love to test. So if you now go back and compare it to the other automation systems that are available, so in our case, the Hamilton system, honestly, I would find it very difficult to say which of these systems is now the better one, because I think they are just completely different. So in terms of size and cost, you can imagine the Hamilton is really a large system that maybe costs more in purchase, and you need a lot of training and experience to get it up and running. But maybe you have also here a higher throughput and flexibility then, because as I said, we're also doing the labeling on that system already. Whereas the Ionic is just really small and easy to handle. It's like a PCR machine that I think every lab can handle, and it's very easy to integrate in your workflow. So I think it really depends on the need of the lab, whether you would go for the one or the other system. They're really complementary to each other, and they're both perfectly fine to work with, at least in our eyes. So with that, I think we can conclude that ITP on the Ionic system now allows us the automated isolation of ultra-high molecular weight DNA that can then be labeled manually or automated on either the Sapphire or the Stratus system. And it is really leading to high-quality DNA, which is less viscous, less fragmented, and is resulting in very good QC values, what we've seen on the Stratus and also Sapphire instrument. Actually, I think that using the Ionic makes it really easier to implement a clinical routine workflow for clinical labs because there's really less user experience needed than you need for the manual isolation. It's much faster due to the 1 hour homogenization step that you need instead of 1 to 3 days. So that's doing a lot on turnaround time, and it's more standardized. And also that is, of course, very important for clinical labs. So based on this, we can conclude that, at least in our opinion, the Ionic system really offers now an attractive alternative for the manual ultra-high molecular weight DNA isolation, especially for clinical labs, but also for any other lab that feels the need to automate. And with that, I'm finished already. I have to thank a lot of people from Radboud UMC, a lot of people from Bionano as well. And I thank you for your attention. Thank you very much.
Thank you to all three of our speakers for a wonderful presentation. So at this time, we do have a few questions for you. As a reminder to our audience, if you have more questions, please submit them using the Q&A function at the bottom of your screen. So first up, for both of our external speakers, one advantage of the Ionic system is that the DNA is processed in a more gentle way, staying in solution. Have you seen that this has led to more consistency in your results?
So yeah, absolutely. I think since the DNA that is isolated is so uniform, and as Dr. Neveling just pointed out, that with the older method, there's shearing, there's dehydration, and then rehydration, and each DNA would be subjected to different exposures. So all of that not being there results in a very uniform DNA, and that results in very uniform QC metrics downstream. And the way these different samples perform all throughout the assay and also on the instrument is very similar to each other. So it does result in very uniform and consistent results.
Thank you. So the next question is for you specifically, Kornelia. What was the starting volume of each sample type and initial cell counts used as input for the Ionic?
Yeah, good point. That was what I nearly forgot to tell, indeed. We used less input compared to Sapphire, and I think that's important to state. For blood, we start with 450 microliter blood, which is usually 750 roughly that we use with the manual isolation. So it's nearly half. And if you start with cell power, then we have 1 million cell count rather than 1.5 that we used to do previously. So we start with less input and still get this very low data, very great data, robust data. And that's exactly the question. What we are asking is how far can we go? And is this really treatable also for tissues or for projects where you really have a low input? And I think there's a lot of people having that need. And I think I do see great potential here, but I would like to go on testing and see from, how far can we go?
Definitely, yeah, thank you.
That is what we're doing now.
So our next question is for Nikhil. You mentioned the one-day protocol. Can you describe how that could be accomplished?
Right. So I really believe that the procedure can be consolidated to 1 day. And again, because the DNA, when it comes out of the instrument, is so uniform, and if you had paid attention, Dr. Neveling pointed out that the CV was 0 or 0.01, or that's the minimum that you can go. So this points out that how uniform or how homogeneous the DNA is. And if you would measure once it comes out of the instrument itself, again, the CV would be very close. So all to say that you can have accurate measurement once you have done the 1 hour homogenization after it comes out of the instrument. And it would be a slightly longer day because you are doing the DNA isolation and the labeling on the same day, but it's certainly possible. And it's important, especially for labs that are looking to make it clinical for hematological malignancies or or tumor samples. So I think that's a real possibility that was not there, at least with the manual method before.
Yeah, thank you. So our next question is for Alex. What were the source materials tested for getting the ultra-high molecular weight DNA?
Thanks, Kira, and thanks, Koni and Nikhil, for the great talks. I think you saw in Koni's presentation that she tested cells, so that would be lymphoblast cell lines, as well as blood and whole bone marrow. Those are the cells that have been most heavily tested internally at Bionano. But we have started looking at some other samples. We're very excited about this technology for the reasons that Koni and Nikhil were just talking about. The uniformity in the output sample quality and homogeneity is really substantial, and so we expect that to enable more tissue types. But we expect to launch the system supporting cells, so cell lines, blood, and bone marrow. And then we'll work on additional kits as we move forward. There was another question in the chat, in the Q&A, that I just addressed as well here, which was whether we've tested with 500,000 cells. And I don't know if Nikhil or Koni have done so, but internally we have tested with lower numbers of cells and have promising results there too. We haven't figured out what the minimum input will be for the commercial launch. We're still working through that.
All right, thank you. So that is actually all of the time that we have. If any of our attendees have additional questions, they can go to the Bionano website, or they can email Alex directly at the email shown on the screen. As a reminder, this webinar has been recorded, so it will be available for on-demand viewing in just a few days. We will send out an email to let you know when that recording is available. Thank you again to Alex, Nikhil, and Kornelia for sharing your work with us, and thank you to everyone for attending today's webinar.